Hello, this is the final part for answering the Unit 1 Molecules, Diet, Transport, and Health for the January 2025 past paper. Okay, so question number 5. Meselson and Stahl provided data that supported the theory that DNA replicates by semi-conservative replication. So, semi-conservative replication. Over here in this diagram, we have one strand, okay? And we have two lines of the red strands. And notice how one red strand goes over here and the other one goes over here. We have the blue colored one, which means it's semi-conservative. So 50% goes here and 50% goes there. So state the meaning of the term semi-conservative replication. So it's one parent strand is retained in each molecule. Replication produces more DNA molecules. This question is always repeated, so make a flashcard for your studies. Now, describe the role of DNA polymerase in the process of DNA replication. So DNA polymerase is an enzyme which adds nucleotides to each new DNA strand. It also forms phosphodiester bonds between DNA nucleotides. Okay, Meselson and Stahl used mononucleotides made with either heavy nitrogen, which is N15, or light nitrogen, N14, in their experiment. The flowchart summarizes part of one experiment performed by Meselson and Stahl. So stage 1, bacteria is grown for several generations in a culture medium containing mononucleotides made with heavy nitrogen N15. Stage 2 is bacteria from stage 1, so meaning that it has nitrogen 15, okay, is grown for one generation in a culture medium containing light nitrogen N14. So basically this is what happens in stage 2. This is what's happening in stage 2, and this is stage 1. So stage 3, bacteria from stage 2, which is this one, okay, is grown again in light nitrogen, okay? So after each stage, a sample of DNA was taken from the bacteria and the DNA molecules separated on a density gradient in a tube. DNA with only heavy nitrogen has a mean density of 7.74. DNA with light nitrogen has a mean density of 7.6. Meselson and Stahl measured the concentration of DNA with different densities. The graph shows the results of these measurements for DNA molecules with only light nitrogen. Okay, that makes sense. Over here, it's at 7.60 and the light nitrogen 7.60. Okay, complete the graphs to show the concentrations and densities of the DNA molecules produced at the end of stage 1, 2, and 3 in the flowchart. Over here, I did a very nice diagram that's pretty helpful. So we have stage 1, it has a density of 7.74, and I gave it the red color. Stage 2, it's grown in the light nitrogen 14. So we're going to have 50% red, 50% blue, or you can say 50% heavy, 50% light. So I calculated the mean density, which is going to be the density of the light plus the heavy divided by 2, which gives us a mean value of 7.67. So we should be expecting a peak at 7.67 in the end of stage 2. And then for stage 3, it's grown again in a light medium. So we're going to have the 7.67, okay, from the red and blue, because the red strand from stage 2 is still present and it's still passed down, okay? And the blue strand is going to join with the blue strand. Same thing over here. So we're going to have a mean of 7.60 and 7.67. So we're going to have two peaks. So stage 1, we're going to have a peak at 7.74, which is over here. And then for stage 2, we're going to have a peak that is double the height of the first peak because we produced 2 instead of 1. So this is a very important point. And the peak for the end of stage 2 is going to be at 7.67. Here's the calculation. For end of stage 3, we're going to have 2 peaks, one at 7.67 and one at 7.60. And here are the calculations and the diagram, which is going to be really helpful. Okay, question number 6. High blood cholesterol level is associated with an increased risk of cardiovascular disease, CVD. The graph shows the relationship between blood cholesterol levels and the coronary heart disease death rate in men in 4 parts of the world. We have Northern Europe, United States, the Mediterranean, and Japan. Describe 2 conclusions that can be made about coronary heart disease. Death rate in these parts of the world. So my first conclusion is going to be the increase in death rate with increase in cholesterol in all countries except Japan. As the blood cholesterol increases, so does the death rate, except Japan. So the second point is the death rate is higher in Northern Europe. That is another conclusion, because here's Northern Europe and it's higher. So that's a valid point. Okay. Cholesterol can be transported in the blood as a low-density lipoprotein, LDLs, that are made in the liver. The diagram shows a LDL. We have cholesterol, ApoB protein, and phospholipids. Statins can be used to reduce blood cholesterol levels. Statins reduce cholesterol synthesis, reduce the synthesis of ApoB protein, and increase the number of LDL receptors on the surface of liver cells. So statins can cause side effects. Give 2 side effects of taking statins. So this is basically asking you side effects of taking medicine. So number 1, headache, and number 2, an allergic reaction. Okay, so question II. Liver cells take up cholesterol by endocytosis. Explain how an increase in these LDL receptors will help to reduce blood cholesterol levels. Well, more LDL will attach to liver cells. Therefore, more will be taken inside the liver cells by endocytosis. Okay. Familial hypercholesterolemia, FH, is a genetic disorder that results in high levels of LDLs in the blood. In the UK, approximately 270,000 people have FH. The disorder affects 1 in 250 people in the UK. Calculate the population size of the UK. So we're going to have to multiply the number with FH times by 250, which gives us 6.75 to the power of 10 times 10 to the power of 7. Okay, the disorder can be caused by mutation in the gene that codes for LDL receptors, suggests why statins are less effective in treating people who are homozygous for this order. Homozygous means same, okay? Because statins work by increasing the synthesis of receptors. A homozygous individual will have mutations in both their alleles coding for LDL receptors. Okay, another drug used to treat FH is a short antisense strand of DNA from the ApoB gene. Suggest how this antisense strand of DNA would reduce the levels of ApoB protein. Well, the antisense strand of DNA will bind to the ApoB mRNA, which will prevent translation, meaning no ApoB protein will be synthesized. Okay, so question number 7. The structure of the cell membrane has been modified to interpret the data available at the time. The diagram shows a timeline of 6 models proposed for the structure of the cell membrane. We have in 1900s the Overton, lipid nature of cell. It just looks like, I don't know, like a slab. Okay, then we have in the around 1915 the Langmuir single layer of phospholipids, and then we have the Gorter and Grendel phospholipid, and then we have the Davison and Danieli protein on the outside of the bilayer, and then we have the Robertson unit membrane consists of dark and clear bands, and then we have the Singer and Nicholson transmembrane proteins showing integral proteins. Okay, so Overton suggested, let's go to the Overton, see Overton, which is over here, suggested that some types of solutes could cross the lipid membrane, whereas other types of solutes could not. Explain the difference between these two types of solutes and why only one type can cross the lipid membrane. So, nonpolar solutes can cross the membrane because lipids are nonpolars. So here, like likes like, okay? Nonpolar likes nonpolar, so it will be able to cross. And polar molecules cannot cross the membrane because they cannot interact with the nonpolar lipid layer. Okay, because the lipid tails are facing inwards, they are nonpolar and are hydrophobic. Okay, Langmuir's model was a single layer of phospholipids. Explain why the phospholipids in the cell membrane cannot be arranged in a single layer. Well, it's not going to be stable, okay? So a bilayer is the only stable arrangement. In biology, it's very important for cells to be stable so that chemical reactions can occur. Okay, so because phospholipids have nonpolar tails that face inwards and are hydrophobic, and polar heads that are hydrophilic facing aqueous environment. Okay, Gorter and Grendel suggested that there were various types of phospholipids, but they were all arranged in a bilayer. Give 2 ways in which phospholipids can be different from each other. Well, first of all, the length of the fatty acids can be different. They can have different amounts of carbons. Number 2, they can be either saturated, means they have single bonds, or unsaturated, meaning they have double bonds. Okay, the Davison and Danieli model was the first to include proteins. Explain why some membrane proteins would not be able to carry out their transport function if they were located only on the surface of the phospholipid. So they're not located through it. They're only located on the surface in one side of the cell. So they're either facing the outside of the cell or the inside. Okay, so proteins, they need to span across the membrane to transport substances because channel proteins are needed for facilitated diffusion. So if we have our phospholipid bilayer, and we have our proteins. If our proteins are facing outwards, here is my protein. So if my protein is facing outward and a molecule wants to enter, well, what's the help in that? So this is not going to work. So our proteins, it's called an integral protein actually. So this allows for facilitated diffusion. Because if it's just on one side, that wouldn't make any sense. Okay, so Robertson implied that there were no space between the phospholipid layers and that the thickness of the membrane could be measured. Oh, actually, I got this picture in my GCSE, so that's a repeated picture, even though this is an A-level paper, but I just wanted to tell you guys. Okay, so the thickness of these cell membranes between X and Y is 22.8 nanometers. Calculate the magnification of this photograph. Express your answer to two significant figures. Okay, so magnification is the size of the image divided by the size of the real. So we have 5.5 millimeters, which is measured by a ruler, and then we're going to times it by 1 million to get nanometers, or you can just write times 10 to the power of 6, which is correct, over 22.8, which gave me 241,228, which I rounded to 2.4 times 10 to the power of 5. Since it's two significant figures, this is my first significant figure, this is my second significant figure. Okay, in 1972, Singer and Nicholson proposed the fluid mosaic model of the cell membrane. Explain why their model has been given this name. This is also a repeated question, so I would advise you to create a flashcard out of this. So it's called fluid because the phospholipids and proteins can move. They don't move up and down, but they do move side to side. So the movement of phospholipids and proteins is going to be lateral. If they move up and down, then it's going to break it and the cell is going to rupture. Okay, and it's called mosaic because the proteins are randomly distributed throughout the membrane. Okay, question 8. Osteogenesis imperfecta, OI, is a group of rare genetic disorders which results in bones that are not formed properly and can break easily. There are several types of OI as a number of different genes can be affected. Some of the symptoms of OI can be life-threatening. The commonest types of OI are caused by mutations in genes coding for the structure of collagen. In some types of OI, the collagen is poorly formed and lacks strength. Explain how a substitution mutation could result in collagen that lacks strength. Okay, so explain what a substitution mutation is first. Okay, so a substitution mutation results in nucleotides being replaced. Obviously now, the mutation could affect one amino acid. This change affects collagen's tertiary structure. Therefore, hydrogen bonds would not form. Okay, this question is repeated in many exams and they might not specifically ask you about collagen, but there is this idea where a mutation is going to change the base sequence or the nucleotides, causing a change in the amino acid, changing the tertiary structure, hydrogen bonds would not form. This question, they could also link it to enzymes because enzymes are also proteins. Okay, in most types of OI, the allele responsible for causing the condition is dominant, meaning it has to be a capital O. Determine the probability that a child could inherit OI if one parent has OI and is heterozygous. Hetero meaning different. So that means one O is dominant and the other O is lowercase. Okay, and the other parent does not have OI. So from the question, we learned that if a parent does not have OI, that means it's lowercase o, therefore it is homozygous recessive and it's given to you in the question. Use a genetic diagram to support your answer indicating the phenotypes of the offspring and their corresponding genotypes. Okay, so the genotypes of the parent with OI is going to be capital O, lowercase o, whereas the genotype of the parent without is going to be homozygous recessive, which is lowercase o. Therefore, when you do the Punnett square, you're going to get a ratio of 50 to 50, or you can say 1 to 1. Remember, they want a ratio, do not put percentages. Okay, now genetic screening. Sometimes they might ask you about genetic screening, sometimes they might not. But it's always important that you have a general idea of the different types of genetic screenings and their pros and cons. Okay, so genetic screening can be used by couples who are at risk of having a child with OI. The genetic screening options include pre-implantation genetic diagnosis (PGD) and prenatal testing. The diagram gives some information about some of the types of prenatal testing available to these couples. We have an NIPT, where a sample of the mother's blood is removed and fetal DNA is used for genetic analysis. It's used between the 7th and the 10th week of pregnancy. We have an ultrasound, which is used a bit later, okay? And it's a scan of the mother's abdomen to look for any fetal abnormalities, and it's usually carried late in the pregnancy, so that's the 20th week. We have chronic villus sampling, which is used between the 10th and the 12th week of pregnancy. We have chordocentesis, where blood from the umbilical cord is removed and then it's gone through the process of genetic analysis. And then we have amniocentesis, which is used between the 15th and the 20 weeks of pregnancy. However, this involves taking samples from the amniotic fluid, which may rupture the amniotic sac, therefore leading to a miscarriage. Okay, so discuss the advantages and disadvantages of the genetic screening options available to couples at risk of having a child with OI. Use all the information in the question and your own knowledge to support your answer. Okay, so the ultrasound would not harm the mother or the fetus, and it would be useful to observe any damage to the bones, because remember, OI, as mentioned in the question, it results in bone abnormalities. Okay, however, the ultrasound can only be used relatively late into the pregnancy. Therefore, it would be difficult to carry out an abortion. An IPT would not cause harm since there is no needles penetrating the tissue. It can be used early on in pregnancy, meaning that abortion would still be an option, therefore there wouldn't be any ethical implications. Now, chordocentesis can be used relatively late into the pregnancy. However, there is a risk of miscarriage if there is any excess blood loss. Thank you so much for listening, and good luck revising. Oh, also, please tell me if you want me to post any other videos or anything else. Thank you so much. Please subscribe, like, comment, and peace.